We will start. Can you hear me? Can you hear me?
Yes, yes.
We will solve June 2023, paper 42. We're still in 2023 versions. Okay, let's start immediately. Figure 1.1 is the diagram of part of a cross section of a leaf. Explain why a leaf is considered to be an organ. Why is it considered to be an organ? Can you tell me what is the definition of an organ?
It's made of tissues.
Okay, made of the same tissues or different tissues?
Same tissues.
Let me see. No, a leaf or an organ is made up of different tissues that
Different.
work that work together to carry out a particular function. 1 minute. So the definition of an organ, this is the first lesson in the syllabus. Don't forget, the organ is made up of different. So why is the leaf an organ? Because it is made up of different tissues that work together to carry out a particular function. Now let's look at the leaf. What are the different tissues that make up the leaf? What is this?
Upper epidermis.
Yes, so the epidermis is one tissue. And what is this?
Palisade sphere.
Yes, correct. And this is another tissue. So this is the mesophyll tissue. And what is this?
Spongy.
This is the spongy mesophyll, correct? This is what's here that carries
The,
the water
the
and the, huh?
vascular bundle or حاجة كده.
The vascular bundle is a tissue which contains the xylem and the phloem. So why is this leaf an organ? Because it is made up of different tissues that work together to carry out a particular function. And give examples. You have the epidermal tissue, you have the mesophyll tissue, you have the transport tissue that contains the xylem and the phloem. Okay, give me a second. Yes, I'll open it from here. Okay, so explain how layer B and layer C in figure 1.1 are adapted for their functions. Now let's go to layer B and layer C. We will talk about the palisade mesophyll and how it is adapted and the spongy mesophyll and how it is adapted. We have a lot of things to say. This question is out of 6 marks. What will you say about the palisade mesophyll?
Many chloroplasts.
Yes. Now this is a core question. A core question that out of 6 marks. So this tells you not to ignore the core material. The palisade mesophyll. Now, the palisade cells, we studied before that these are specialized cells. These are long to increase their what? They are long and rectangular to increase their,
Surface area. No.
correct, to increase their surface area to carry a lot of photosynthesis. They have many chloroplasts to carry out lots of photosynthesis. They are tightly packed, as you can see. They are arranged so that they are just below the epidermis, so that they are close to the sun. They are at right angles to the sun. Now, this is the palisade mesophyll. What about the spongy mesophyll? What will you say about the spongy mesophyll?
It has air spaces.
Correct. They have lots of air spaces. And what is the advantage of those air spaces?
Surface area برضه.
Also, correct, they increase the surface area for gas exchange, for the circulation of gases. And they also have chloroplast for photosynthesis. And what is this part?
Vascular.
Yes, you are correct. Now, what will you write? This vascular bundle, what does it contain?
Xylem.
Correct. It contains the xylem and the phloem. And you can discuss the structure of the xylem and the phloem and how they are adapted. This question, look at this question. It is out of 6 marks. So you will write everything you know about the palisade mesophyll, the spongy mesophyll, the even the transport bundle, because you can see it in the spongy mesophyll. So you will talk about the xylem and you will talk about the phloem. You can talk about the xylem in specific because this is clearly in your syllabus. It's made up of hollow tubes on top of each other and strong lignified walls, no obstruction, no cytoplasm, so that the water will travel smoothly, no nucleus. Okay, now I want to look at the mark scheme. No, I'll tell you exactly where will you find it. This is year 9 notes. We want to look at the features of the palisade cell. Okay, they're here. So you'll find the features of the palisade cell on page 22 in year 9. The palisade mesophyll or the plant cell function photosynthesis. They're long or column in shape in order to increase the surface area, provide maximum photosynthesis, lots of chloroplasts to trap sunlight, positioned right below the upper epidermis. They're tightly packed. This is about the palisade mesophyll. The spongy mesophyll, like we said, they're irregular and they have lots of air spaces for circulation. And you will talk about the xylem in detail, everything we took about the xylem, and you talk about the phloem. The xylem, okay, this column about the xylem. You don't have to write the whole column, but at least talk about the structure. The xylem dead, no nucleus, no cytoplasm, hollow tube, end-to-end, degenerated end walls, etc., etc. Let's look at the mark scheme. This is which question? This is question 1. We're still in question 1. Here. So you will talk about the palisade mesophyll, and you will refer to photosynthesis and the chloroplast, and then you will talk about the spongy mesophyll. This is, we're still in layer B. Cells are tightly packed. This is palisade. And immediately below the upper epidermis for light energy, for chloroplast, we said all this. And then we'll talk about the spongy mesophyll. The cells are loosely packed with many air spaces. We also said that for gas exchange. And you will talk about the structure of the xylem. And also you can say what is the function of the phloem. So when you have a question that is out of 6 marks, now you have to worry. I always tell you that. A question out of 6 marks that you have to worry. You first need to label layer B and layer C, and you will discuss them in detail. Layer B, you will talk about the palisade mesophyll. You will talk about everything you know about the palisade mesophyll structure and function and how it is adapted. You have a nice paragraph here in year 9 notes. How is the palisade mesophyll adapted to carry out its function? And then you will discuss the spongy mesophyll in detail. And then you will discuss the xylem in detail if you want to take the 6 out of the 6. Okay, this is the core question. A student placed a plant in a very hot room for 12 hours. There was a bright light in the room, and the plant was not given any water during the 12-hour period. Figure 1.2 shows a series of sketches that the students made of the stomata during the investigation. Now, please concentrate on the investigation. The room was very hot for 12 hours, and there was no water for 12 hours. So what will happen to the guard cells and the stomata? What is happening with time? Can someone tell me what will happen to the guard cell? What
Closes.
is D actually? Identify D. What is D?
Guard cell.
The guard cell. And what is the main function of the stomata?
Uh, gas exchange.
Very good, gas exchange. State the advantage to the plant of the change to the stomata shown in figure 1.2. Now we can see at the beginning that the stomata is open and then the stomata is closing. Now, why is this an advantage? Why did the stomata close?
Prevent water loss from
Reduce water
transpiration.
loss.
Yes, to reduce transpiration because we don't want loss of water. The students increased the humidity in the room and repeated the investigation. Predict and explain. So this is a two-part question. The effect of high humidity on the stomata. And this is 3 marks. Now predict. What do you think will happen to the guard cell? Will it close down like this? Or it will not close down, it will stay open?
Close.
Why? We said we will increase the humidity. It closed down because it was very dry and very hot. So we closed the guard cell to reduce transpiration. Now we increased the humidity.
شترت.
So the guard cell will close or will remain open.
Open.
Yes, the guard cell will not close quickly, or it will remain open for longer periods of time. Okay? Now explain. Why will the guard cell remain open? When the guard cell, this is the guard cell. Now, these are the questions where you lose a lot of marks and you don't understand the question. The guard cell is surrounded by a humid environment. Humid means what? There is high water potential. There is high water potential outside, and there is also high water potential inside. So the potential gradient or the potential difference is not high. The amount of water inside the guard cell is close to the amount of water outside the guard cell. So will the guard cell lose water
Gain.
and become flaccid and close?
It lose water.
No, it will not lose water. You forgot. Now we took the opening and the closure of the guard cells, we took it in transport in plants. Let me show you. Here. The guard cells, when do they open and when do they close? You completely forgot, and your exam is only after 1 month. The guard cell, they will collect water, or they will, this is the guard cell here, I cannot draw here. The guard cells, they will, let me open a screen. Okay, the guard cell is made up of two cells. You have two guard cells that are kidney in shape. When do they open and when do they close? When water enters into the guard cell, they curve outwards and they open. And when does the water enter the guard cell? When the guard cell is doing photosynthesis, water will enter and it will make the guard cell turgid. The vacuole of inside the guard cell will be filled with water and it will become turgid and it will curve outwards. Now, when does the guard cell do not open? Looks like this. When the guard cell is flaccid, when it loses water, when the water leaves the guard cell by osmosis, and the vacuole will shrink and it will wilt or it will become flaccid and it will close. So we said that the guard cell will be surrounded by a humid environment. It will be very humid. This means that the guard cell will not lose lots of water by osmosis. The water potential inside the guard cell is close to the water potential outside the guard cell. The water gradient or the concentration gradient will decrease. The difference in concentration of water inside the guard cell and outside the guard cell will decrease. So less water will leave the guard cell by osmosis. That is why the guard cell will remain open or turgid for a longer period of time and it will not wilt, it will not close. Do you understand this question?
Yes.
Okay, this question is very important. And something like this, you will not be expecting that they will leave the whole syllabus and ask you about the guard cell, but they just did. Okay, now let's move on. So all this question is about plants. Figure 2.1 is a diagram of part of a DNA molecule. Draw a circle on the figure to identify one pair of bases. This is easy. This is a base. I expect you to know it. So you will do one circle. The percentage of thiamine bases or T bases in the DNA is 29%. Calculate the percentage of bases that will be base G in the DNA of this species. How will you calculate that? What is the complementary of base T?
A.
A, very good. So if T is 29%, what is A?
61, 71.
My dear, A and T will be the same percentage.
Oh,
29%.
So if T is 29, what is A?
29.
29 also. What is the total? A and T, this is 29 plus 29, 58%. 100 minus 58%.
42%.
This is 42. And then we'll divide 42 between base C and base G. So 42 divided by 2, this is 21%. The answer is 21%. Who does not know how we got it? Okay, let's continue so we finish. State the name given to a length of DNA that codes for a protein. That is easy. Hmm?
Gene.
Yes, it's a gene. Now explain how proteins are made by a cell. Again, this is a direct question. You studied it, good for you. You did not study it, you lose 5 marks. Who will remind me what are the 2 steps needed in the making of protein? You don't know the answer and your exam is in 30 days. I don't believe you.
Transcription and translation.
Yes. So discuss what is transcription. Transcription.
This is the messenger RNA copy a part of the DNA.
Very good. The DNA is copied on a messenger RNA. Please say that the DNA does not leave the nucleus because it's too big. And then that's it. This is what you will write about transcription. Okay. Not all the DNA, part of the DNA that codes for the gene that we want. What is the second step? You said translation. What happens in translation?
Messenger RNA moves to the ribosomes through the cytoplasm.
Correct. It will go out into the cytoplasm. It will attach to our ribosome. And what will the ribosome do?
Gets translated. The genes get translated onto the ribosome.
Yes. How do we do it? We said it will arrange the sequence of.
Amino acids.
Correct. So we will translate the messenger RNA, and there's a very popular sentence that we write. It will assemble the amino acids, and the sequence of amino acids, the order of the amino acid depends on the what? Sequence of bases on the messenger RNA. This is a very important sentence. It is bold in red. And this sentence should always be in the answer. I'll show you. This is, we said question 2. Okay. The ribosomes make proteins or polypeptides. We already know this. Messenger RNA is copied from the gene. The gene stays in the nucleus. This is important. The nucleus or the messenger RNA moves to the cytoplasm. Messenger RNA passes through our ribosome. We said that ribosomes will assemble the amino acid or translate the messenger RNA. The order of the amino acids is determined by the base sequence of the messenger RNA or the DNA or the gene. This last sentence is very important. Don't forget it. DNA controls cell function by controlling the production of proteins. State 2 types of cell membrane proteins. What are types of proteins that can be found on the cell membrane? Now this is easy. What protein can be found on the cell membrane that will receive a hormone or a neurotransmitter or what will you write? You will leave
Can
this
we
question.
say enzymes?
Yes, of course.
Or
Enzymes can be
structure?
proteins and receptors is a protein. Antibody is a protein. You have lots of proteins. Okay. Now graphs. I know that you do not like graphs, but you will have to receive a graph in paper 6 and paper 4, whether we like it or we don't. 3A, antibiotic resistance is an increasing problem worldwide. Erythromycin is an antibiotic. Figure 3.1 shows the daily doses of erythromycin per 1,000 people over a 13-year period. The number of bacterial infections resistant to erythromycin per 1,000 people is also shown. So look at this graph because this graph has lots of information. It has a key where it will tell you that the dark color is the daily dose of the antibiotic per 1,000 people, and the light graph is the number of bacterial infections that are resistant to this antibiotic. On the x-axis, you have the years. On the y-axis, you have different information. You have the dose of the antibiotic. And on the other side of the y-axis, you have the number of bacterial infections resistant. And you have the dark and you have the light bars. Okay? Now, the first question, calculate the percentage change. in the number of bacterial infections resistant to erythromycin per 1,000 people between 1993 and 1995. Who will remind me of the formula?
Final minus initial over initial times 100.
Correct. Minus initial over initial times 100. So the first thing we do, we want to do the final and the initial. So the final will be 1995 or 1993?
1995.
Correct. So we'll go to 1995. They want the percentage change in the number of bacterial infections. So don't get confused. We will use this or the values on this side, the number of bacterial infections. And which bar? The light bar or the dark bar? Look at the key.
The light bar.
Correct, the light bar. So this can cause lots of confusion. Just be careful. So we will go to the light bar and the final value will be 100. So the final value will be 100. And the initial will be 1993. I'll go to 1993. I'll go to the light bar. The value will be 180. So it will be 100 minus 180 over the starting divided by 180 divided over the starting times 100. Now the value or the answer we will receive will be negative or positive?
Negative.
Negative. And it's logic because the values, they did decrease from 1993 to 1995. Don't be scared when you get a negative answer. 100 minus 180 equals minus 80 divided by 180 times 100. Now I got on my calculator minus 44.44. The question is telling us to give my answer to two significant figures. So what do I write?
Negative 44.
Correct. Now, please be careful that you have space for working. So what we did here, you do it here. You have to show them how you got the answer. Don't just write minus 44 and that's it. There are marks for the steps. Let's see if we are right. Yes, minus 44. Correct. Okay. So percentage change is important. Now describe the data shown in figure 3.1. This question always makes you lose marks because you do not describe the graphs in detail. We want to describe this graph. This is not an easy graph, so please highlight it so that you revise it later. You have this graph with years on the x-axis, with the daily dose of erythromycin on the y-axis, and also on the y-axis, the number of bacterial infection, and you have two bars. Look, I will give you an advice. When you come to describe a graph, look for number one, the trend, if it is increasing or decreasing, and always discuss the peak or the maximum value. So let's look at the dark bars. The dark bars, which represent the daily dose of erythromycin per 1,000. What is happening to the dark bars from
Increase and
1988?
reach their peak at
Very
1980.
good. Very
Then
good. This
decrease.
is excellent. This is how you write it. So the trend is that initially, or from 1983, they are increasing until they reach peak at 1989 and right values. The peak is, if we are talking about the daily doses, the peak will be around 2.8 or 2.7. Okay. And then, like your friend said, they will start decreasing until they reach this value in 1995. Very good. Now let's talk about the other bar. The number of bacterial infections. Do we have any values in the first few years?
No, it starts appearing at 1991.
Correct. So you will say that, and then the bars, they increase, and then they
Decrease.
decrease, and there's a peak value at 1993. And write the value. If you are talking about the number of bacterial infections, give a number. If you're talking about the daily dose of erythromycin, give a number. Okay? This is going to be a very good way to describe the graph and take the 3 marks. You wrote everything, and the peak and the increase and the decrease. And also you can say the relationship between both of them. 1991, they're nearly the same. 1993, this bar or the bacterial infections increased way beyond the other bar. So you can say a lot of things. Suggest reasons for the change in the number of bacterial infections resistant to erythromycin from 1993 to 1995 in the figure. So I'll have to first go to 1993 and look at the number of bacterial infections, which is the light bar. I can see that there's a decrease. So the number of resistant bacteria the number of bacteria infections resistant to the antibiotic. So the antibiotic resistance decreased, not increased. Can you suggest reasons for a decrease in the antibiotic resistance?
Since the dose of it increasing as well is decreasing as well. Sorry.
Excuse me, my dear.
Since the other column, the darker column, is also decreasing.
No, this is not a reason. Why did the antibiotic resistance decrease? Usually, the antibiotic resistance increases with time with overuse, but in the graph, we see that the antibiotic resistance decreased. Why? Suggest. Suggest means a critical thinking question, a new question out of syllabus. Can you think of ways why the resistance to antibiotic decreased? Ask yourself, why
People,
would the resistance to an antibiotic increase? We took it because of our good habits or bad habits.
people use them when they don't need to.
Yes, because of our bad habits. Now, this question is the opposite. The antibiotic resistance decreased. So we stopped using or stopped doing the bad habits. We stopped using it in unnecessary situations. We stopped using it in viruses. Maybe new antibiotics have been made. We became cleaner. We did antibiotic sensitivity tests before using any antibiotic. We did not use it for viral infections. We used the correct dose. We only use it when we need it. So here, it's the opposite. We have bad habits that increase the resistance. But we see from the graph that the resistance decreased. So we stopped doing those bad habits. Do you understand the question? Okay. We did not use it except for viral infections. There is improvement in the hygiene. We became cleaner. The bacteria did not spread. We used new antibiotics. We used the antibiotics only when necessary. We did not use the antibiotics with livestock. I think you know what livestock is with cows and sheep for unnecessary or when it is not necessary. We use the antibiotic in the correct dose. We isolated the patients that are sick so that we do not spread the infection. Okay? We don't need to look at the mark scheme and waste time. Explain how the bacteria become resistant to antibiotics. How many marks? 5 marks. Now here we're talking about what? Who will remind me? How does the bacteria become resistant to the antibiotic?
Mutation leads to variation.
Excellent. You will write the whole story. Can you start, please? First, like you said, there is mutation that will lead to what?
Variation. And,
Correct.
and then some antibiotic become resistant and reproduce.
Stop. Some antibiotic or some bacteria?
Bacteria, sorry.
Yes. Huh. Some bacteria,
Bacteria
correct, perfect, will become
become
resistant, and then some will not be resistant. Correct. And then what else?
the resistant will have traits and reproduce more, leading that the non-resistant will die
Correct. The non-resistant will die.
due to competition.
They will
Due
die. Okay, because they are not resistant. And then this will reduce competition or increase competition.
when they die, this reduces the competition, so
Correct.
the resistant reproduce more.
Yes,
And
perfect. So
there is no
the resistant bacteria will start multiplying and producing more, and their resistant genes will be passed on. Yes, don't forget that. Until all the bacteria will have the resistant gene. All the population of bacteria will evolve to have the resistant gene. And this is an example of what the most important word.
natural selection.
Yes, correct. Natural selection. What was the selective pressure and what was the survival advantage?
The survival advantage is the being
I did
resistant
not hear you. The survival advantage is the what, my dear?
is them being antibiotic resistant, I think.
Correct. The resistant gene. You're right. I want you to be sure of your answer. And what was the selective pressure?
Stuff like um,
What made them die? Like what made the nature choose which bacteria will resist and survive and which will not?
um,
The antibiotic, the presence of the antibiotic. Okay, are you with me? The antibiotic is the
Yes.
selective pressure. It is the pressure applied on the bacteria that will make either those, some will die and some will not. So this is a long story that we took it in variation and selection, and we took it again in ecology, and we took it in inheritance. It's actually, and we took it in the lesson drugs. Okay, it's in actually in 2 lessons at least. And this is 5 marks, so don't forget it. Bacteria are prokaryotes. Now, this is the first lesson, cell structure. State 2 features of all prokaryotes. What are the features of bacteria? I think you know it very well.
No membrane bound organelles.
Correct. Give me an example. What's the
Uh,
only organelle they have anyway?
ribosomes. I think.
Correct. Perfect. They only have ribosomes. Give me another feature.
Have
Uh,
plasmids,
Yes, correct. They have plasmids.
cell wall of peptidoglycan or
Yes, they
murine.
have a cell wall. They have no nucleus, and their cell wall is made up of peptidoglycan. Very good. And they have no nucleus, and they're small, and they have no organelles. Perfect. Now, the bacteria have a flagellum. What's the function of the flagellum? That's so easy.
Allow movement.
Hmm?
Help it move.
Correct. Movement or swimming or movement. Very, very good. It's an easy exam, by the way. But some questions are tricky. Now, an ecology question. You always have an ecology question. So please don't ignore ecology. A student recorded the information about an aquatic habitat. What does aquatic mean?
Live
Live in
underwater.
water.
Yes, correct. Figure 4.1 shows the student's notes. Aquatic plants provide food for freshwater shrimps and midges. Mayflies also feed on aquatic plants. The mayflies provide food for salmon. Stoneflies feed on midges and are eaten by salmon. Salmon also eat freshwater shrimps, and herons feed on salmon. Okay, this is a food web. Construct a food web to show the feeding relationship described in figure 4.1. Do not draw the organism. Now, this is tricky question. But what will you do? You will simply follow every single sentence. Let me show you. So we have, of course, any food web or food chain starts with a producer. So we will start with aquatic plants. So I'll write here, aquatic plants. It provides food for freshwater shrimps. So I'll make an arrow. It is eaten by the shrimps and midges. Okay, so I drew the first sentence. The mayflies also feed on aquatic plants. So I can do this. The aquatic plants, they are fed or they're eaten by the mayflies. Or I can do another food web. I can write another one, a separate one. Aquatic plants, like a separate one for every sentence, is eaten by the mayflies. Both are correct. And then I finish the second sentence. The third sentence, the mayflies provide food for salmon. So the mayflies are feeded by, they're eaten by what? Salmon. Or I can go back to my second one and write mayflies eaten by salmon. They're both correct. Okay, so this is the third one. The fourth sentence, stoneflies feed on midges and are eaten by salmon. Salmon, let's do sentence by sentence. Stoneflies feed on midges. So the midges are eaten by stoneflies and are eaten by salmon. So the stoneflies are eaten by salmon. Do you understand what I'm doing? And the last sentence here, salmon also eat shrimps. So the shrimps are eaten by the salmon. Herons feed on salmon. So the salmon is eaten by herons. Herons is the final consumer. Did you understand how we did it? This is not an easy question. Also, I want you to highlight it. This question is asking you to draw a food web. Usually they give you the food web, but this question, they asked you to draw it. They gave you the order of the plants and what they eat, and they asked you to draw it. And what you will do in a question like this is that you will follow sentence by sentence. I'll do it again for you. Plants is food for the shrimp and the midges. So this is what I did. I wrote the plants they're eaten by the shrimps and the midges. Do you understand the first sentence? Any questions? The mayflies feed on the plants. So these plants are also eaten by mayflies. This is the second sentence. The mayflies is food for salmon. So the mayflies will be eaten by salmon. Check mark. Stoneflies feed on midges. So I will go to midges. They're eaten by stoneflies. And these stoneflies are eaten by salmon. So the stoneflies are eaten also by salmon. Salmon also eat the shrimps. So the shrimps are eaten by the salmon. Herons feed on the salmon. So the salmon will be fed or will be eaten by herons. Who does not understand this food web? Okay, no one is asking me, so I expect you all to understand it. Complete table 4.1 using the information by identifying the names of the missing trophic levels and one organism at each different trophic level. So you have to draw the food web properly so that you are able to answer this question. What is the primary consumer? Give me one primary consumer. Let's go to my food chain. Give me one primary consumer.
The shrimps and midges.
Yes, you are correct. So the shrimps and the midges is the, or can be one and midges. Primary consumer, correct. Secondary consumer. Also, it's easy.
Stonefly.
Yes, stonefly. This is the producer. This is a primary consumer. This is the secondary consumer. So the stonefly will be the secondary consumer. Okay. Now, we want the name of the trophic level. What is after the secondary consumer?
Treachery.
Correct. Tertiary consumer. And then the quaternary consumer or the top consumer. You will continue the table. Let's go look for a tertiary consumer. I want a tertiary consumer. Primary, secondary.
Salmon.
Salmon. And also a top consumer. It is also so a tertiary consumer, salmon. And it is also the top consumer. It is also salmon. Let's look at the mark scheme. Okay. We forgot the herons. There's a heron. Oh, we forgot the heron. So the heron is the top consumer. So if you don't do your food web correctly, you will not be able to answer this question. But we did it correctly, and we did not just see the heron because the picture is too big. Let's go look at the mark scheme again. We drew the food web, okay? It is very similar to what we wrote, but it's just neater. The producer, the primary consumer, we said the shrimps, correct. Secondary consumer, the stoneflies or the salmon. Tertiary, the salmon or the herons. And the quaternary consumer, the herons. Who did not understand this question? This question is not easy. Always there's a question that is tricky. So this question, in my opinion, if you don't know how to draw the food web, you will not get it. This is the producer, primary consumer. Also, the mayfly can be a primary consumer. Stonefly, secondary consumer. Salmon, the tertiary consumer. And the herons is the top consumer. Any questions? Do you want me to repeat it for you, or is it clear? Okay. Outline how the energy in the primary consumers in this aquatic food web is used to produce biomass in the secondary consumer. Now, again, this is year 9. Talk to me about the primary consumers and how do they trap energy. This was the first sentence in the lesson. Who will answer this question for me? How do the primary consumers use energy? This energy first, it is absorbed by what? It is, how is it transferred? Who will tell me? I'm not optimistic. Hmm.
The energy is gained by consuming the produce.
Okay, first let's go back to the very, very beginning. Who is the principal source of energy? Who is the main source of energy? This is year 9 material.
The sun.
The sun. And this sun energy is going to be absorbed by the
Producer.
producer in photosynthesis. And the producer will change the sun or the light energy into
Chemical energy.
correct, into chemical energy. Okay, look at this question. How does the energy move through an ecosystem? The energy will enter an ecosystem as sunlight. The producers will absorb the light energy and convert it into chemical energy, which is food. And then this chemical energy inside the producer will be transferred from one consumer to the next as they are being eaten until finally the last organism will die and it will be decomposed. And you will also mention this food is used in what? In growth, in respiration, in mitosis, in production of proteins, etc., etc., etc. Now, I honestly do not expect you to remember. So this question also highlight it, please, because it's an important question. So please highlight this question and go back to it while you're studying. Outline how the energy in the primary consumer in this aquatic food web is used to produce biomass in the secondary consumers. So we will write what we just discussed in the year 9 notes. First, we have the main source of energy. So the energy will arrive from the sun. It will be absorbed by the plants in photosynthesis, and it will be converted into chemical energy. Okay, this chemical energy will be transferred from the plant into the primary consumer. And then when the primary consumer is eaten by the secondary consumer, it will be transferred and the chemical energy will be transferred and so on until the top consumer. How will it be transferred? By ingestion. It will be eaten. Now, how will it be converted to biomass? This chemical energy will be broken down in what? The food will be broken down in how is food broken down? Photosynthesis or respiration?
Respiration.
Yes. So the food inside the bodies of these animals will be broken down in respiration and it will give us energy. And this energy will be needed or be used in growth, in making proteins, in cell division, in creating biomass, gaining weight. Okay? I expect you to say this part of the answer. You will all know or remember that the food will be broken down in respiration, but I don't think you will remember the first part of the question because this is year 9 notes. So please do not ignore also year 9. Let's go to the marked scheme. Any three from the primary consumers will contain chemical energy. So the chemical energy is important. This energy will be transferred to the secondary consumer and so on from the primary to the secondary, blah, 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 as they are eaten. We said that. The food or the primary consumers will be digested to produce energy. We said it. From respiration, energy is released and used for growth and to make more cells and mitosis, for protein synthesis, for reproduction, for assimilation, stored to create biomass. So we said everything, but I do not expect you to remember the first part of the question, which is the part of the chemical energy. Okay, so this is written here 9 in your year 9 notes. How is the energy moving through an ecosystem? Please do not ignore year 9 notes. Sometimes the question is a combination of 9 and 10, like this one. Humans also eat salmon. Predict the impact on the feeding relationship shown in figure 4.1 of over-harvesting of the salmon. So let's go back to our food chain. Imagine that the salmon is gone, no salmon. How will this affect the remaining organisms? Can you tell me?
The mayflies and stoneflies and the shrimp and midges will increase.
You are correct. So the mayfly will increase because it will not be eaten by the salmon. And the stonefly will increase because it will not be eaten by the salmon. And also the shrimps. Now, what about the herons? Will they find food?
Decrease because
Yes,
they
they
die.
will decrease. Yes, they can become extinct because there is no food. So this is going to affect the herons. They will decrease. There will be no food. And the other organisms, they will increase. Okay. And when the other organisms increase, for example, the stonefly, this is going to eat more midges. The midges will be eaten more because I will have more stoneflies. Okay. Also, when the mayflies increase, they will need more aquatic plants. So you just need to go back to your food web and look at the relationship. This is easy. Okay. Describe what is meant by decomposer. What is a decomposer?
An organism that feeds on dead organism.
Yes, it feeds on dead organic matter. Correct. C. Animals such as salmon can be farmed for meat. Explain why it is more energy efficient for humans to eat crop plants than to eat livestock that have been fed on crop plants. Now, I don't know if you remember this part. This part, we took it here. I don't know if you understand the question anyway. Here,
Because the energy is
It's due to
transferred only
energy.
10%.
less energy lost. Remember this? Think of the following food chain involving the humans. And this is also in year 10 notes. 1 minute. The problem is there are so many details. You study all of them, but I know that you will forget. So this is what is bothering me. Okay, here. What if humans eat the wheat? Why is it if humans eat the wheat, there is more energy available to them than if they eat the cow? So we said always that when the humans are the herbivores, this is much better. When the human eats the wheat directly, look at the first food chain, we have less trophic levels. The human here will eat the wheat. So the human will receive more energy. But in the second food chain, the human is the secondary consumer. The wheat is first eaten by the cattle, and then the cattle will be eaten by the human. The second food chain, we said it is less energy efficient because more energy is lost. Every time the energy is transferred from one trophic level to the next, we have loss of energy in movement, in respiration, in excretion, and lack of digestion. So when we have more trophic levels, we have more loss of energy. We will give examples how energy is lost. Why? Less energy is being lost because there are fewer links in the food chain. Links means trophic levels. Less energy is lost in a food chain with fewer trophic levels. So hopefully you will remember this. I don't know. You need to study really well and study all the details. So it will be more energy efficient for the humans to eat the crop plants because in this case, the human will be the herbivores. And the food web will include only two trophic levels, the plant eaten directly by the human. So I will have less trophic levels and less heat loss or less energy loss or more energy loss. Wake up with me. Less loss of energy or more loss of energy?
Less loss of energy.
Yes. In that case, it will be more energy efficient. Again, this is not enough. This is out of 4 marks. How is the energy lost? How much percentage of energy lost is lost at every trophic level? Who will remind me?
Only 10% is passed on to the next consumer.
Correct. Correct. 10% of the energy is passed on. And how is it lost?
By heat or exercise
By
or
movement, by heat, by excretion, by undigestion. Not all the food is digested. Okay, because we want the 4 marks. Good. Let's look at the mark scheme just to be sure, but this is definitely correct. Uh, it has fewer trophic levels. They are at a lower trophic level. Plants are at a lower trophic level. Energy transfers along the food chain is inefficient. We said that energy is lost in respiration, heat, metabolic processes, blah, blah, blah. More material is lost in feces, in excretion. Okay. So you give examples of how energy is lost. This exam doesn't finish. The last question. Figure 5.1 is a picture of a structure found in animal and plant cells. What is this organelle? The one with lots of foldings to increase the surface area. For
Mitochondria.
aerobic respiration, come on,
Mitochondria.
mitochondria, yes. So the name, this is mitochondria for aerobic respiration. State the two pieces of information needed to calculate the actual length of cell structure A in figure 5.1. So figure
image length
5.1, we want the actual length. What is this is easy. What is the formula? I am They want the actual length, so they want the image size, millimeters, and the magnification. Are you all with me? So the actual length of the cell structure is 0.0075. Convert this value to micrometers. This is so easy. What do you do?
Multiply by 1,000.
Yes, 0.00075 times 1,000. And they already gave you the units. This, okay, it doesn't finish. Now, a student investigated plant growth responses in roots and shoots. They used this method. This is coordination. Damp cotton wool was placed in 2 petri dishes. 3 bean seedlings were attached to the cotton wool. Each seedling was oriented so that the roots pointed in a different direction. Petri dish 1 was kept on its side in a fixed position, so this was fixed. Petri dish 2 was kept on its side and rotated constantly. Both petri dishes were kept in the dark. So here we are studying geotropism or phototropism if it's in the dark.
Geotropism.
Geotropism.
Geotropism, correct. Both petri dishes were kept in these conditions for 2 days. After 2 days, the seedlings were observed. Now we have this picture exactly as it is, and we took it before. So this is petri dish 1. This is stationary, it is fixed. And this is petri dish 2, and it is moving. This is rotating. What is this device called? The rotating device? Clino? Oh,
Shy.
clinostat. The clinostat that keeps on moving and moving and moving in these experiments. And why does it move? It acts as the what? Oh my goodness.
As the constant, the control.
The control. Yes, it is the control to cancel out the effect of the gravity or the sunlight. Here we are studying gravity. I don't know what to tell you. I don't know what to tell you. It's right there. Where did it go? Here. Exactly the same picture. To act as the control. Exactly the same. Exactly the same. As if they took copy and paste. Okay, now the petri dish 1, these are the results. All the roots now they are pointing downwards, as you can see. Let's see, what do they want? Describe the pattern of growth of the bean roots and shoots in petri dish 1. So go to petri dish 1. What will you say about the roots?
Roots
How
are
did they
facing
grow?
downwards.
Yes,
They
and we have a name. This is positive what?
positive gravitropism.
Correct. This is positive geotropism or gravitropism. Now, what about the shoots?
They grow upwards, and this is negative geotropism.
Correct. Perfect. So this is negative geotropism. Easy. State the name of the growth responses observed in the bean roots and shoots. We just said it. This is geotropism or phototropism.
Geotropism.
Correct. 3. Explain how oxen causes the differences in the pattern of growth shown by the shoots of the seedlings B and E shown in figure 6.2. Go to B and E. B and E. Now go to B. The shoot here, the shoot, they're asking about the shoots. It will point upwards. So this is the shoot. It was horizontal, and then the shoot will point upwards. Now we discussed this 101 times. Can you discuss to me the story of the oxen?
The oxen forms at the tip of the shoot,
Correct.
and
It will
then
be produced, and then it will move by.
diffusion to
Correct.
the lower side of the shoot, the one facing
Correct. The side towards gravity, and then
this
it will stimulate.
stimulate growth on the shaded side.
Yes, it's called cell elongation. Don't forget the word. It will cause growth on the shaded side and cause cell elongation, and this will make it bend towards the
The
away from the gravity. Correct. And this is equal or unequal distribution of oxen.
unequal distribution.
Unequal. Perfect. Now, what will you say at E? Why did it grow straight? Don't forget the petri dish 2 here is rotating.
It was rotating constantly, so The oxygen diffuses equally on both sides.
Perfect. Excellent. Okay, this is a cleanostat. It is a control to cancel out the effect of the rotation. Perfect. This is 5 marks. Don't please miss it out. Seeds require oxygen and water to germinate. State one other environmental condition that affects germination. Remember the WOW? So they said oxygen and water. What is the last W?
Warm
Warmth.
Correct. Temperature. Suggest why oxygen and water are required for germination.
water is to activate the enzymes, I think.
Correct. Water to activate the enzymes, to transport the material, to dissolve the sugar. All the chemical reactions take place in water, any one of those. And the oxygen for what? For
Uh,
which type of respiration?
aerobic
Aerobic.
respiration.
Yes, for aerobic respiration. Now, I want you when to read the question, always the question is usually a two-part question, like this one. Suggest why oxygen and water are needed for germination. So, right, oxygen is needed for XYZ, and water is needed for XYZ. Be organized because usually you're not, or you write one and you forget the other. The last question. Two examples of protease enzymes are pepsin and trypsin. Pepsin, oh, I forgot the first one. Enzymes are involved in chemical digestion, which produces small what molecules?
Soluble.
Correct. Soluble. Okay. Two examples of protease enzymes are pepsin and trypsin. Pepsin is produced by the
Stomach.
Stomach.
stomach. Correct. And requires acidic conditions. These conditions are created by the release of what?
Hydrochloric acid.
Perfect. This is digestion, which provides the optimum pH for pepsin activity and also kills harmful.
Microbes or bacteria.
Yes, correct. Microbes or bacteria or pathogens. The what produces trypsin?
Small intestine.
Small intestine.
Correct. The small intestine, or you can write also the pancreas, which breaks down protein in what conditions?
Alkaline.
Alkaline.
Alkaline. Correct. Alkaline conditions. Well done. These conditions are created by a substance called, who will provide the alkaline medium?
The bicarbonate.
Yes,
The
sodium bicarbonate in the bile. Correct. You are perfect. Which will neutralize the gastric juices and also has an important role in the what of fats and oils?
emulsification.
Emulsification. It's an easy question. We finally finished this exam. This exam is not difficult. It's an easy exam. In my opinion, the questions that were difficult, the oxen question is a direct question. You studied it, you will find it, or you will do well. You did not, it's your problem. Okay. The food web. So I want you to highlight this question, the food web question, this one. Okay. Repeat it and do it alone on a paper and do the food web. And also this question, the energy sufficient one. So I'm going to highlight it for you. This one. Okay. I can send you this version of the exam to show you the questions that we thought were difficult. So we will highlight this question also. How bacteria become resistant to antibiotics? It's easy, it's direct, but if you did not study it, you will lose all the 5 marks. And there was a question out of 6 marks. The graph was not easy. Always the graph is not easy. So describe the data shown. We will discuss the trend. Let me write it down. The trend of both graphs and the peak and the relationship. Good. And also the question, the first question out of 6 marks, and also the guard cells. The guard cells. And how do they function? I'm sure you will forget it. This question out of 6 marks, how layer B and C are adapted for their function. It's very easy, but this is a year 9 question. That's out of 6 marks. This is year 9 syllabus. You will discuss the palisade mesophyll, spongy mesophyll, and the xylem structure. The xylem structure is year 10. So this question is combining 9 and 10 together. Okay. Any questions before we go? Please solve as much as possible. And any questions you have, send me on WhatsApp. I'll answer you. Clear? Okay, I'll send you this exam or the mess we made to show the difficult questions, and I'll meet you on, we said Friday 2 p.m. Bye bye.